Hello, my name is Daniel Movsesian, and alongside my lab partner Maria Khramsova, we are going to be talking about the development of a plant-based versatile culture medium supplemented with cabbage and incorporating purple cabbage as a pH indicator. In recent years, there has been a notable shift in the scientific community towards ethical and sustainable practices, driven by a deep understanding of responsible resource use and a commitment to eco-friendly innovation. Our experiment aligns with this ethos, contributing to reducing environmental impact in laboratory research by proposing overlooked components from food and agricultural industries, such as damaged vegetative buds, outer leaves, stems, thereby reducing waste reduction. And creating a cabbage-based medium not only promotes sustainable lab practices, but also aligns with ethical considerations, supporting animal welfare and respecting diverse dietary, cultural, and religious choices. Our vegan approach in laboratory practices reflects a commitment to ethical scientific methods that account for the environmental impact, animal welfare, and cultural considerations. Now let us explore why cabbage and red cabbage make excellent components for our vegan-based media. Cabbage is a nutrient powerhouse for bacterial growth, with sugars in the form of polysaccharides acting as an energy source, supporting metabolism, and facilitating growth in a culture medium. Rich in essential minerals and water, cabbage provides critical elements like carbon, nitrogen, and magnesium, ensuring an optimal environment for bacterial growth and new cellular synthesis. Remarkably, cabbage contains 1 to 2 grams of protein per 100 grams, which significantly contributes to the overall nutritive value and the ability of bacteria to proliferate when added to a medium. Red cabbage serves as a valuable purpose, acting as a natural pH indicator due to a compound contained within them called anthocyanins. These pigment-rich chemicals, responsible for its vibrant color, uniquely respond to changes in pH, offering a non-invasive method for identifying shifts within a growth medium. Now, our exploration of vegan-based media uncovers a historical reliance on animal-derived products in laboratory culture media, dating back to Louis Pasteur and Robert Koch. Traditional sources like coagulated egg parts, milk, diluted blood, meat, and urine were all common nutrients for bacterial growth. While studies explore vegan product compatibility with mammals, soytones, tryptones, and vegetable infusion powder from potato, soybeans, peas, or tomatoes now dominate media production. However, cabbage has primarily been used simply for kimchi starters and hasn't been used in this specific context. Therefore, recognizing this knowledge gap, our experiment aims to contribute to plant-based media evolution. Egyptian scientists have emphasized using cactus and succulent plants for cultivating rhizobacteria, and this is a notable advancement in developing general-purpose plant-based media.
So for our project, we have two main objectives. The first one was to create an all-purpose vegan media that is capable to support healthy growth of gram-negative and gram-positive bacteria. And the second one was to create a pH indicator from purple cabbage and to test its compatibility with the cabbage media. The hypotheses for our study were the cabbage media provides all the necessary nutrients for bacterial proliferation. And the second one, the cabbage pH indicator, demonstrates the sensitivity corresponding to the colorimetric scale that we created, and it can serve as a substitute for commercially available pH indicators.
Our experiment strategically includes a diverse panel of microorganisms to comprehensively evaluate the adaptability and efficacy of our vegan-based media. Variations in microbial morphology from gram-negative bacilli to gram-positive bacilli and fungi allow us to assess how different microorganisms respond to vegan-based culture medium, providing insights into their adaptability. This selection also showcases diverse metabolic pathways, reflecting the metabolic diversity of gram-negative and gram-positive bacteria, including organisms associated with foodborne pathogens and opportunistic infections, enhances the ecological relevance, while the use of fungi broadens our understanding beyond simply bacteria.
For culture maintenance, we obtained master plates, and we chose one isolated colony from the plate and streaked it for isolation on TSA, incubated it at 37 degrees Celsius for 24 hours, and afterwards we just kept it in the fridge for further usage. So media preparation from a cabbage included the following steps. The first one, we had to wash and cut the plant matter. The second one, we processed it with a blender. In order to obtain the slurry, we added the water to it. Afterwards, we added the cellulase and pectinase enzymes to the slurry to break the complex components. We waited for 12 hours. Afterwards, we strained our slurry. We divided the solid part and a liquid part. The liquid part we boil in order to precipitate peptides that are present in a cabbage juice. And in our previous experience, after autoclaving, they appear as a sediment or as a clumping. So after boiling, we had to additionally filter it in order to obtain the clear media. After boiling, we measured the pH of our cabbage juice. It was 7, so it's neutral pH. We didn't have to adjust it with the acid or with the base. And afterwards, we added the agar powder, boil it, and make sure the agar is absolutely dissolved in our cabbage juice. And after, we send it for autoclaving, and after autoclaving, we distribute it between plates and tubes. Cabbage pH indicator was prepared in 3 different ways. The first one is sock slat extraction, where dehydrated cabbage leaves we extracted with the help of a distilled water. The second one was a hot water extraction. Technically, we immerse the chopped cabbage in a boiling water for 10 minutes in order to extract the pigments. And the third one was to immerse the filter paper in cabbage juice and then dry it in order to create a pH indicator strips.
Now, we took steps to ensure the sterility of our materials through a negative control. Media and buffer underwent rigorous sterility checks. After a 48-hour incubation at 37 degrees Celsius, no bacterial growth was detected on TSA plates, cabbage media, or the phosphate buffer. The absence of this growth serves as a robust negative control, which affirms the reliability and precision of our experimental setup. In our exploration of cabbage-based agar media, we conducted 2 pivotal tests to gauge its effectiveness. Now, firstly, we assessed the media's suitability for bacterial and fungal proliferation among selected microorganisms. This test was crucial in understanding how well the medium supports microbial growth. Simultaneously, we undertook a second test to optimize the concentration of cabbage extract in the media. Fine-tuning this component is essential to create an environment that is conducive to microbial development.
Figure 1 demonstrates the results of strict plating of our microorganisms of interest after incubation at 37 degrees Celsius for 48 hours. They exhibited the robust growth on our plates. After incubation, we collected all our data and put it in the table 1 in order to demonstrate the intensity of growth and the change in the color of the media. Another part of our testing was to prove that cabbage media can support the healthy bacterial growth even while being diluted. So for that, we plated E. coli on plates with a different concentration of cabbage extract, and as it demonstrated in figure 2, cabbage media can support the healthy growth even while being diluted. Another important requirement for the media in general is to be clear and transparent. Especially the liquid media. For that, we created the cabbage broth and then inoculated it with the microorganisms of our interest and kept 1 tube as a control. And after incubation, the tube with inoculation demonstrated the sediment and turbidity, which proves the bacterial growth inside of the tube, and the control tube stayed clear and without any sediments. For pH indicator testing, we use liquid cabbage broth that we inoculated with our microorganisms and incubated it at 37 degrees Celsius for 24 hours. Afterwards, we checked the pH inside of the tubes with the pH indicator strips. And then in order to check our cabbage indicator, we added 5 drops to each tube. And as you can see our results in a figure 4, our microorganisms produce different level of acidity due to their metabolic activities. Based on our pH indicator strips results, we created a colorimetric scale that is helpful for interpretation of the results. As demonstrated in Table 2, we compare the results for our pH indicator and the commercially available pH indicator, and as you can see, the difference is very insignificant. After conducting all our testings and collecting all our results, we can confirm that we created versatile vegan media that is suitable for growth of different microorganisms, such as gram-positive and gram-negative bacteria and fungi. Also, we proved that our media can support the healthy bacterial growth in different concentrations present in the media. When we speak about the liquid cap media, it didn't exhibit any clumping or sediment or cloudiness in uninoculated control tubes, which proves that our media meeting another requirement. And on the other hand, the tube that was inoculated with the microorganisms, it demonstrated the cloudiness and clumping, which confirms the bacterial growth inside of the tube. And lastly, the cabbage pH indicator exhibited a vivid color change according to the acidity of the broth, which occurred to the metabolic activities of the bacteria. Based on that result, we created the colorimetric scale, which can help in future to interpret the results for pH indication.
Now, in conclusion, our study aimed to innovate by formulating a natural, vegan, and versatile cultural medium capable of supporting the growth of diverse bacteria and fungi. Simultaneously, we sought to utilize a natural alternative to chemically derived pH indicators. The obtained cabbage medium demonstrated its efficacy in providing ample nutrients for both bacterial and fungal proliferation. This marks a significant stride towards developing sustainable and plant-based alternatives in microbiological research. Furthermore, our exploration into a purple cabbage-derived pH indicator showcased promising results, positioning it as a potentially effective substitute for commercial chemical pH indicators. The following slide showcases the references used for this experiment. Thank you for watching.